So I'm very happy to introduce our speaker today, John Grealy. John's a professor in the Department of Genetics and Pediatrics at Einstein, and also the director for the Center for Epigenomics. He grew up in Ireland, did his training at the National University of Ireland in Galway, also trained at the Children's Hospital of Pittsburgh and Yale. It does research, as his title would suggest, in understanding the non-coding genome, function of non-coding variants, and its relationship to phenotype. And so, thanks so much, John, for joining. And yeah, very excited for your talk.
Thank you, Kyle, for the introduction. I'm going to start off with something which is perhaps a little bit unusual. I want to acknowledge the virtual visit to the land of the Kumeyaay, to the recognition that our native brothers and sisters have suffered genocide and theft of their land, and their ongoing displacement remains an injustice. And also that my economic privileges are in part resting on the history of enslavement of people in the United States with this legacy of anti-Black racism. So hopefully you'll have some things that you'll take with you as regards the scientific content of this talk today, but I'd also ask you to consider these ongoing injustices as well. The map that you see here is kind of reflective of injustice in its own way. The very pale blue at the top is the Bronx, and this is an income map from a few years ago. I work in the most diverse county in the United States, and the South Bronx is the poorest congressional district in the United States. So one of the things that I'm passionate about in our big gleaming healthcare facility here is how we deliver healthcare effectively to individuals of all backgrounds and circumstances. And one of the things that I'm unusually fortunate to be able to do is to be able to practice genomic medicine in a way that's pretty unconstrained. And the reason is there's so few patients in our health system who have commercial insurance that we effectively, as an accountable care organization, pay for our patients' testing. So I don't need to get pre-authorization from an insurance company to be able to order an exome or a whole genome. And the problem is that I'm living in this privileged position, and I recognize just how bad we are at medical genomics. The return that we get for sequencing everybody's genes or everybody's entire sequenceable genome is about maybe 20% of my patients, and that's not good enough. So that's the clinical problem, and that is one of the reasons why I want us to be exploring the non-coding genome to understand what might be going on there, contributing to human phenotypes. So here's examples of some of the more positive studies, and as you can see, the positivity rate, even in these selected cohort studies, which are designed to give you higher diagnostic rates, we range between about 12 and 38%. And over at Rady Children's Hospital, they probably have a higher number these days than 19%, but at the time, this was what we had for whole genome sequencing of critically ill infants. But the bottom line with this is that you're sending away most of your families without a diagnosis. And this is despite the fact that we have all of the sequence of every gene or the entire genome. And this we've got to do better. So the more real life example is this study that I was involved with, the NYC KidSeq part of the CSER consortium, where we were looking at individuals from diverse backgrounds as defined by the NIH, who had cardiological, neurological, or immunological presentations. And as you can see, although there were some differences in the sites here between what we were getting in Montefiore and Einstein versus Mount Sinai downtown, only about one patient out of 5 was getting a diagnosis, and 4 out of 5 were sending home without diagnosis. So this reinforces the need for what I wanted us to be studying. And here's the problem. We only really diagnose coding sequence variants. What you have here is a genome browser view from UCSC. And in red are pathogenic and likely pathogenic variants. Blue is variants of uncertain significance, and green are benign or likely benign. And as you can see, we're only really calling this diagnostic information in exons. That we don't call information from the non-coding regions. And this locus was deliberately chosen because it's the imprinted region on chromosome 11p15 that's responsible for Beckwith-Wiedemann syndrome and Russell-Silver syndrome. And it's a really well-studied location. There's KCNQ1 again there. And over here, you can see the really unusual gap in the high C map, which is the very well-known H19 insulator. This is a non-coding sequence which is involved with setting up imprinting of the IGF2 gene. We actually know a lot about non-coding sequences, but we just don't translate that over to medical use in an effective way. So look at that, isn't that beautiful? So what do we think about when we think about the non-coding genome? We basically are talking about a regulatory landscape, and we have to use these so-called epigenomic assays to be able to understand where these regulators are. What you're looking at here is the time course experiment where we differentiate mesenchymal stem cells into basal medium, and then day 3 and day 7 of osteogenic differentiation in vitro. And as you can see, some of the peaks are beginning to become stronger or are appearing de novo, while others are shrinking as you go down this pathway. But these are very related cell types where one is derived from another. If you were to look at tens of cell types, you'd start to realize that the regulatory landscapes are actually quite complex in their differences between the different cells and tissues. So we refer to these genome-wide studies of transcription regulation as epigenomic studies. And now that says, well, that we're making some assumptions about epigenetic regulation, and I need to take you down this back road here and give you a little bit of a briefing on how we should be thinking about epigenomics. And so bear with me on this. And I'm hoping that this book, we've got a mocked up title page for it here, that this book will come out in 2023. I had a lot of fun writing about the whole area of epigenetics, but I realized the only way of writing it was to anchor it in history. So I found it very instructive in terms of where we are today as a field. And I want to bring you through that history very, very briefly so that you don't have to buy the book. So here's the problem. In the early part of the 20th century, the embryologists had just got over the fight between the people who felt that there was a homunculus that was the pre-existing structures and they just needed to get bigger from the egg or the sperm, versus the epigenesis individuals who posited that specialized structures were formed from non-specialized looking precursors. And of course they were right. And it was of course the development of the microscope that allowed people like Wolf to show this. But here's the problem. Early part of the 20th century, people started to irradiate animals. And they started to see that there were these kinky tails. This is the T hairpin mutation in the upper of the mice here. And as you can see, the fly on the right does not have red eyes. So that by itself is, maybe there's some damage, but what they were showing was that these were heritable phenotypes. So the geneticists started wandering over to the labs of the embryologists and said, isn't it interesting how the heritable material is actually involved with morphogenesis? And the embryologists, who were very upset that their turf was being invaded, they said, listen, you ignorant people, it's exactly the same they knew about chromosomes. There's the same chromosomes at the front end of the mouse as the back end of the mouse, or the left side of the fly as the right side of the fly. So how could that be deterministic in any way? It can't possibly be doing anything for development. Please go away and learn some science. I can show you some stuff where they were equating the geneticists with using language for describing Nazi Germany. It was really over the top. So Waddington, the famous Waddington who came up with the word epigenetics in the first place, pro tip, if you want to look serious, smoke a pipe. Waddington's point of view was uniquely informed. He was a geology PhD dropout who ended up dabbling in biology and got the chance to work with Hans Spemann in Germany, who was a classically trained embryologist, and it was the person who described the Spemann organizer. But he also had the opportunity to go out to Caltech on a traveling fellowship and work with two of the former Columbia Fly Lab members, Alfred Shortevant and Theodosius Dobzhansky, and was able to see what they were doing with these new genetic approaches. So he had a foot in both of these camps, and he was the why can't we all just get along person. And his idea of the epigenetic landscape was a combination of epigenesis, or cell differentiation, and cell fate determination and genetics. So this very famous diagram, which you've seen before, is probably saying less than it has subsequently been interpreted to say. All he wanted to say was that cell fate was the ball choosing one or other canal or creode, as he called it. But on the what was holding the landscape in place were these guy wires, and the guy wires are genes. So if you snap a guy wire, you'll change the conformation of the landscape. So this is gravitationally implausible, because for the upper diagram, you need gravity to be working from below so the ball will roll down the hill. But for the lower diagram, for some reason, gravity needs to be lifting up this landscape so the guy wires will actually do something. So ignore that and just get with the idea that Waddington was proposing that genetic influences on cell fate could be the way that you could reconcile the two warring fields of embryology and genetics. So the model deservedly just didn't go anywhere. And then David Nanny, a tetrahymena researcher who was really interested in the idea that heritable information could be transmitted in the cytoplasm of tetrahymena, specifically mating type locus information. And he was absolutely right, as it turns out, because siRNA mediated mechanisms in the cytoplasm are actually what mediate the memory of mating type. So he was going to a meeting of these heretics who didn't believe that everything was encoded in the cell nucleus. So he was flying from Indiana, and he took a 14-hour propeller plane ride across the Atlantic, got to Paris, got down to the southwest of the city where the meeting was on, and was sitting down for dinner the night before he was due to give his presentation. You have to imagine the guy was absolutely shattered from the traveling. So he sits down beside a fellow called Guido Pontecorvo, and Pontecorvo says, "What are you going to be talking about tomorrow?" And Nanny says, "I'm going to be talking about paragenetic inheritance of this information about mating type loci in tetrahymena." And Pontecorvo, who had grown up in Italy and studied Greek, classical Greek as a young man, said, "You can't call it paragenetic. That's wrong from a Greek point of view. You have to call it epigenetic." And they said, "Oh, that's Waddington's word. I can't use that. Waddington's meant to be at this meeting. Let's talk to him." Waddington didn't turn up. So because Nanny was so intimidated and so jet lagged, he ended up using the word epigenetic to mean cellular memory. And that was a complete reinvention of the word, born of necessity. This is the only picture you're ever going, well, one of the only two pictures you'll ever see of John Pugh. Excuse me. John Pugh was a graduate student working in the lab of Robin Holliday. I suspect that John was not that focused, because while he was meant to be working on Holliday junctions and recombination repair, he ended up becoming fascinated by X chromosome inactivation, Mary Lyon's hypothesis of the random inactivation occurring early in life and being maintained. He went to watch a pair of seminars by Ruth Sager, who was over in London, talking about her work on restriction modification systems, in which she was talking about how the modification of methylation of DNA in bacteria would protect against restriction enzymes. And this was a light bulb moment for John, who realized that if the DNA methylation could be in some way maintained from parent to daughter cell, that this could be a mechanism for the memory involved implicit in Mary Lyon's hypothesis. So this is John with the long hair here, second from the left, pictured in Heidelberg. And this is John in 2019, just before the pandemic, when I tracked him down in Bethnal Green in East London. And we had a conversation where he told me how he came up with the word epigenetic, which is shown here in this abstract. As he said, he had a 200-word limit to discuss something about DNA methylation and how the gain or loss of DNA methylation could be a mutagenic event, which was Robin Holliday's interest. And because of his 200-word limit, he needed a word that encompassed this idea of something which was added to the DNA. So he came up with the word epigenetic. He had never heard of David Nanny, and he had never heard of Hal Waddington. So if you were to, first of all, word limits are a curse, because this is what ends up happening. But I think if you were an evolutionary biologist, you'd say that the evolution of the word epigenetic has followed convergent pattern. In other words, it has evolved separately many times without relationship to the prior events. And I would like to pay tribute to the late R. Riggs, who came up with exactly the same ideas about DNA methylation and X chromosome inactivation based on restriction modification systems when he was working in the Boyer lab. And he had the good grace not to refer to it as epigenetic, never did. So this is the history of the word epigenetic. Now let's ask ourselves the question, why do we think that epigenetic processes mediate disease? And this is where we end up talking about mice and assays and misinterpretation. So a very influential model is the mice that I'm depicting at the bottom here, shown in the last slide, which is the viable yellow mice. They have a retrotransposon called the IAP element that has inserted upstream from the non-agouti gene, which is involved with coat color. And when you have the constitutive activation of this gene, you end up with yellow hair. The phenotype of a litter is a mixture between the very yellow mice, the mottled animals, and the ones that look like the animals that don't have a mutation at all, which are called pseudo-agouti. So the second part of the phenotype is that the animals with the yellow fur also tend to be obese and have metabolic syndrome and have a reasonably good representation of the type 2 diabetes phenotype. So what George Wolfe did was he gave them this high single carbon donor diet and was able to show that, as depicted by the arrows here, he was able to shift the proportion of animals that looked either mottled or pseudo-agouti to a higher proportion within each litter, with fewer of the animals with the yellow fur. And as it turns out, this was associated with DNA methylation and silencing of the IAP element. So now, this is the ultimate guilt trip for pregnant mothers, right? Because you're talking about an environmental exposure during pregnancy that changes the risk of a child going on to become an adult with type 2 diabetes. And so this, again, had an epigenetic feel to it because of the idea of memory being propagated over time. And it involved DNA methylation, which is a heritable mark from parent to daughter cell. So we started to have this idea that extrinsic influences of the environment could be transmitted to chromatin, DNA methylation, and so on, and could influence the regulation of these marks in the genome, such that you would have the loss of chromatin accessibility, the gain of DNA methylation, and the silencing of the gene, or vice versa. So let's think about now how we think of this in terms of tissues and cells. So you go from being healthy, or you go from being unexposed to exposed, and now you've got this phenotype, this disease, this effect of the exposure, and I'm just depicting it in the liver here. We're assuming that the cells have changed, that the cells have picked up new characteristics, and that these new characteristics are in turn due to the change of a molecular genomic phenotype at loci in the genome, typically many loci in the genome, that interfere with normal gene expression. And that's why these cells are different. That's why there is a disease. So I would refer to this as a reprogramming of cells. However, here's the problem. This finding on its own, when you go in and you want to test your epigenetic hypothesis and you see these changes of DNA methylation, gene expression, chromatin state, whatever it might be, these observations are fundamentally uninterpretable because of the fact that they may not represent the reprogramming that you're looking for. So on the right here, I show a blue cell type in which the cytosine of interest is methylated, whereas the equivalent cytosine in the orange cell type is unmethylated. And if you've just got a lot of cells that are blue, the locus will look very methylated overall. But if you flip that ratio, the locus will start looking less methylated. But here's the problem: no cell has actually changed their methylation, there's been no reprogramming going on. The other thing that we have begun to realize is a major influence on DNA methylation, chromatin structure, and so on, is DNA sequence variation. And what you're looking at here is, it's just one of many studies. This one is from our group, where you've got a SNP distinguishing the parental alleles, a blue and a red. And if you've got the red SNP, everything nearby, or most of the cytosines nearby, are methylated, and vice versa for the blue SNP. Now you may say, well, maybe this is just an imprinted locus where the maternal allele is methylated, paternal unmethylated, and so on. But this is genetic, because whenever you get the blue SNP inherited, whether it's from your father or your mother, you always get a lack of methylation nearby, and vice versa for the red SNP. So we don't look for these things when we are testing these epigenetic hypotheses for disease. We try to do cell subtype proportional changes, but our heuristics for that are pretty crude. And there's almost never any genotyping done looking for DNA sequence variant effect. But here's another issue, that we're probably studying transcription factor biology, because you have to ask the question, why was this locus chosen in the first place to change its chromatin structure and DNA methylation? What was the sequence specificity that was involved there? You don't want the enzymatic mediators of DNA methylation and chromatin states to have any sequence specificity, because they need to work at different places in different cell types, right? So they need to be brought by the hand. They're visually impaired, and they're told, come here and acetylate histones, or come here and perform TET oxidation of methyl cytosine, whatever it might be. So DNA, it can be targeted in a sequence-specific way by transcription factors. And what transcription factors do is they invite their friends. They've got these activation domains, and they're able to recruit histone deacetylated remodeling complexes, whatever it might be, so that you can have these local changes in a very site-specific way. So let's think not in terms of autonomy of decisions by these so-called epigenetic regulators, but that when we see these changes, they're probably footprints of transcription factor activities. Now, John Stamatoianopoulos in the University of Washington published this study. I have all the PubMed IDs over here on the left. Early in the pandemic, and I think it was ignored unfairly because of that, because everyone was distracted thinking that we're all going to die. But what John was able to show was in hundreds of different cell types, not only where these open chromatin regions were located, but because he used DNA's hypersensitivity, he was able to see the relative protection of the sequences where the transcription factors were binding. So John asked the question, what's the sequence variation at these loci in human beings where the transcription factor is binding? So he looked at the TopMed data, looking at tens of thousands of individuals, and he quantified that variation at the transcription factor binding site. And the way that it's going up here is a reflection of how these transcription factor binding sites between us all are relatively more polymorphic than the flanking sequences of DNA. And what he was also able to show, looking at the DNA's hypersensitivity patterns, was that half the time there would be a decreased affinity of the transcription factor, and half the time an increased affinity. So we have to think of our transcription factor binding sites as being in a kind of a Goldilocks state, where they can, a sequence change is as likely to make them more avidly binding to the transcription factor as the possibility that they will reduce their ability to bind. So now, thinking in terms of transcription factors, you can ask legitimately the question, well, what regulates transcription factors? So there are two major mechanisms, as you all probably know. One is that they're nuclear receptors. Something binds to them, and then they translocate from the cytoplasm into the nucleus, and they find their target sites. But the other is that they live downstream of cell signaling pathways. So if you can infer from the loci in the genome that are changing well, let's break it into two. On the left here, we have loci that are doing something different following your exposure. This could be DNA methylation, chromatin states, whatever, gene expression. And what we tend to do is try to find a biological coherence of all these loci, and we do gene ontologies or protein-protein interactions, and we come up with these kinds of representations of the biological coherence of these loci. I would argue that that's a secondary effect. That's after the genome has had its chance to respond, it comes out with this type of response. But if we want to understand what is the primary mediation of the environmental influence, if we can use the transcription factor motif information to infer the transcription factors that are getting activated or inactivated, we can track back to the likely cell signaling pathways that we know regulate each of these individual transcription factors. And this is more like the primary response to the environmental perturbation, and another way for us to be thinking about how we could be analyzing our data in a better way. So when you have this kind of change here, it could be that it's cellular reprogramming, which is typically the hypothesis that we go in with. But it could also be cell subtype proportional changes, DNA sequence variant effects, altered cell signaling mediated through transcription factors. And this is why I think hypothesis-based research in genomics is a bad idea. Because, and this is this very interesting commentary by David Glass, who now works at Regeneron, is well worth a read from the point of view of philosophy of science. Because as David points out, if you're wedded to an hypothesis, you can only fail if you don't generate data that supports the hypothesis. So you will be biased towards trying to make your hypothesis fit your data. But if you instead ask a question, you say, what is associated with this change here? And you're open to these other alternative mechanisms, which you would otherwise have dismissed as confounding effects. Then with this question-based approach, you are open to really interesting pathophysiological insights. And I would encourage people to think along those lines. So now this is the way that I've kind of rethought what we should be doing with these epigenomic assays and trying to understand the introgenic space. So how did we apply these insights? I want to show you a few vignettes, relatively superficial, but to give you a sense of how these things can be used in your research career. Hang on a second while I just clear my throat. Okay. So this is work by three graduate students from my group, Neetha Olahan, Rihanna Donia, and Ronnie Cutler. What they did was a very simple experiment where we took toxoplasma and infected human foreskin fibroblasts, which is a classical model for how to study toxoplasma. If you leave the cells too long, the toxoplasma will basically kill the human host cells. But after 24 hours, the cells are still very viable. They look very healthy. So at that point, we did RNA-seq in bulk and ATAC-seq in bulk. And for the bioinformatics people in the audience, you make a combined genome so that the alignments can go natively to each of the genomes, and it doesn't force toxoplasma alignments onto the human genome and so on. So beautiful data set. What did we find? So you look just a few of the results here. We had a ton of stuff in this paper. Gene expression is mostly upregulated, and the most upregulated gene of all is this matrix metalloprotease. Now, this is the host RNA that we're looking at here. And what we saw was that a few of these metalloproteases, matrix metalloproteases, were upregulated. Now, this is not advantageous to the host cell. These are enzymes that tend to chew away at the extracellular matrix and allow the cell to slide from where it was to somewhere new. It helps the infection to spread. So that made us think, should we be looking to see how the toxoplasma is co-opting the host transcriptional regulatory machinery in order to be able to help itself? So when we looked at this cloud of loci here, we realized that as well as immune response, which is very much advantageous to the host, it's trying to do something to get rid of this pesky infection. It also had a cell division response, which is well known in the toxoplasma literature, and a metabolic response. Now, the metabolic response is really interesting because toxoplasma is what's called auxotrophic. It cannot make its own nourishment. It can't make its own metabolic requirements. So it has to use host pathways. And this is clearly advantageous to the toxoplasma. So we asked the question, what are the transcription factor binding motifs that are overrepresented in the host genes that are upregulated? And as you can see, we've got an AP1 response, NF-kappa B, and this RAS-related transcription factor as well. So when we looked at these loci that were uniquely being hit by these specific transcription factor groups, all of them were associated with the immune response. But the NF-kappa B response was restricted to the immune response, which makes a lot of sense for those who are NF-kappa B experts. But as you can see here, the AP1 is associated with cell division metabolic changes, while the RAS-related pathway was associated with metabolic and extracellular matrix changes. So these are some fascinating insights into how the toxoplasma this may be evidence for how the toxoplasma is co-opting the host transcription regulatory pathway. So we know that there are some mediators from toxoplasma that influence host cell signaling. What we would be interested in, given the fact that we've implicated AP1 and the RAS-related pathway, is what toxoplasma mediators are influencing ERK and MAP kinase signaling that could be upstream of these transcriptional responses. So again, a different way of thinking about how we look at genomic data to infer cell signaling. Here's a very simple example of something different, which is a change in a proportion of a cell subtype in a phenotypic example of aging in this case. So we have a very large study that we're in the process of moving into the analytical phase on. It's very exciting. It's 200 people per group, young and old, and we've got CD4 positive T lymphocytes, and we're doing single cell RNA-seq on all of them, and attack-seq and genotyped, and it's going to be this big, hairy data set that we hope will be informative about the physiology of aging. But this is a preliminary study. We just did 6 individuals per group, 3 male, 3 female. And as you can see, there's a group of cells here which is higher proportion in the younger individuals, and a second group of cells which is lower proportion in the younger individuals and is higher proportion in the older individuals. It should be absolutely no surprise to anybody that these represent naive T cells that are reduced as you age, and cytotoxic T cells that increases your age, which reflects the fact that with time you end up being exposed to more antigens and your repertoire changes accordingly. So we know that with aging there are changes of DNA methylation and gene expression. This is a little lesson that says we need to consider that some of these changes are going to be due to these cell subtype effects, and this is a representation of these naive and cytotoxic cell proportion changes with age. And I'm really looking forward to being able to tell you what we find when we have all 400 of the samples feeding into these assays with integration. Now, I love this study. It's my colleague Maria Kundakovic from Fordham University in the Bronx. And what Maria was interested in was the idea, that it was unfair for the neuroscience community to be excluding female animals because of some supposed variation of behaviors that was due to the estrus cycle. What is known, and she's decided to make it a positive model for her studies. And it's a really interesting model because when the mouse is in a low estrogen state, she's very tentative and does not explore an open field and so on, all of these neurobehavioral tests, as you can see. But when the estrogen level goes up, the anxiety, the timidity decreases, and they become much more exploratory of their environment, much more courageous. So very interesting phenotype. When you take the ventral hippocampus, which according to experts like her is the part of the brain that should be mediating this, the spectacular thing that you see is that dendritogenesis of the neurons in this region changes over the 4-day period. You get dendrites being formed in the high estradiol state and then regressing over this 4-day period. So the cells are actually undergoing some changes themselves. We did nuclear attack-seq on the neuronal cells from the ventral hippocampus. You're going to get some New York ambient sound in the background for a second, fire truck going by. And we were able to see changes of chromatin in the genome in using a TAC-seq, where some loci were closing and others were opening over this 4-day period. And this was associated with gene expression changes nearby, going up and down. So an extraordinary dynamism of chromatin regulation. So we, again, had this transcription factor-centric idea. And we said, what are the transcription factor motifs that are overrepresented? And what you will not see is the estrogen response element. You would expect that if it's an estradiol-mediated event, that you would see that. We didn't. What we saw instead, which was informative, was, apart from the MEF2 transcription factors, was EGR1. And EGR1 was very interesting because we know something about EGR1. We know that when the estradiol binds to the membrane-bound estrogen receptors, it triggers a MAP kinase cell signaling response that ultimately mediates its activities through EGR1, getting into the nucleus and acting on the target cell, the target loci. Whereas we did not see the estrogen response element, as I said, so it was not a nuclear receptor version of the estradiol estrogen receptor that was mediating this effect. So again, insights because we were thinking in terms of cell signaling. Here's another example, and now we're getting into toxicology. So there are some endocrine disrupting chemicals that are in our water systems that cause sex reversals in wildlife, and some of them are obesogenic. And the one that we decided we would study was tributyltin, which is 3 butyls and a tin. It's basically pretty self-explanatory. Tributyltin causes increased adiposity in everything from C. elegans to mammals. Its mechanism has been thought to be due to its binding to RXR and PPAR gamma and acting as a kind of an agonist of its activities, thereby driving adipogenesis, because PPAR gamma is like the master adipogenic regulator. So we did, Taylor Thompson and the group did this differentiation, regular differentiation. You're looking at gene expression variation here on this principal components plot. We studied rosiglitazone, which is a known PPAR gamma agonist, because we thought that this would replicate the tributyltin effects, and it really didn't have much of an effect at all. But when we looked at tributyltin, it had early, sustained, and major effects to reprogram these cells. They no longer had the characteristics of straightforward white adipose tissue. They were kind of beiging, and you had hypertrophy and hyperplasia, as well as this very significant amount of gene expression reprogramming. So, cut a very long story short there, we didn't find any evidence whatsoever when we did our transcription factor motif analysis that suggested that RXR or PPAR gamma was in any way being increased in its representation or binding affinities. What we did see instead was that there was decreased TEAD transcription factor, TEAD transcription factor binding from early and sustained through, and that we were starting to see a lot of activation of the RAS-related GTPases like Rho GTPase, CDC42, and so on. So both of these were sending us a consistent message. The RAS-related GTPases are involved with the maintenance of cytoskeletal integrity, whereas the TEAD transcription factors live downstream of hippo signaling, YAP-TAZ signaling. And they get, when the cell has a nice stiffness to it, the TEAD transcription factors are able to bind to their target loci in the genome, and they maintain a osteogenic or whatever state. But if you lose that TEAD activity, then you start having induction of PPAR gamma programming, and that allows the cell to go down an adipogenic lineage. So the primary evidence that we have here is that there's probably some damage to the cytoskeleton. We found some old data suggesting this to be the case from papers in the 1990s. And we've started to study this with phalloidin, fluorescent phalloidin assay, and we can see that there's an actual loss of cytoskeletal beta-actin in these cells in the dosages of tributyltin that were used in the study. So this again was we went in there believing one thing. We used the transcription factors and the cell signaling information, and we came out with a completely different model for how this stuff is working. Okay, now we're getting to the final part of the talk here. And this is where I'm going to start talking to you about DNA sequence variation. And this is hopefully going to be the biggest takeaway of the talk today. Excuse me. So I was chatting with Joe Gleason, and we were talking about this new approach that we have been developing, which we call, because we have to call it something, Regulatory Landscape Enrichment Analysis, RLEA. It's kind of got the same sort of vibe as GSEA, so people hopefully won't get too scared by it. But this is the idea that if you've got some loci of interest, and I'm just putting it here in terms of GWAS loci, and you want to understand what's their relationship to regulatory elements in the genome, what you can do is you can take a bunch of cell types, and there's now a TAC-seq on tens of cell types available publicly, and you can ask whether you have any overlap of your loci of interest with the TAC-seq peaks in each of these cell types, and you can then test to see whether through permutation or other analyses whether this overlap is exactly what you'd expect by chance, or whether it's enriched or depleted in some way. So this is a package that Sam in the group is currently bundling up. It's written in Python. It'll be a command line interface, and it should be something that we can distribute fairly shortly. So getting back to Rihanna, we decided we would use this to try to understand osteoporosis. She had taken mesenchymal stem cells, and she had looked at the early differentiation up to day 7. With in vitro differentiation down the osteogenic lineage. And as you can see, a huge amount of change of gene expression, of TAC-seq patterns between mesenchymal stem cell state and the basal medium. And then in the osteogenic medium, another big jump by day 3. And it then sort of becomes a lot more placid in terms of how it goes forward with fewer changes by day 7. So our question was, these osteoporosis risk loci, are they affecting the mature osteocyte, or are they influencing the way that we may be generating these osteogenic cells in the first place? And what was in my head was, when you look at these osteoporotic bones, you've replaced the trabeculae with basically adipose tissue. And so me thinking in terms of MSCs, I was thinking that, this is maybe a choice, and these variants could be associated with this Waddingtonian bifurcation, and which cell type you end up destined to create. So we took the 2600 loci from GWAS and we overlaid it, and the green here is really to highlight how in the MSCs, day 0, day 3, and day 7, we were unbelievably enriched. The Z-scores were like 17, 15, and so on. So these are very non-randomly overlapping, these MSC and early osteogenic TAC-seq peaks. And about 10% of all of the 2600 loci were ending up in these peaks. So this looks pretty amazing. Our question then was, is this something which is only seen in these cells? So we put another 50 cell types in. I'm just showing an example of GABAergic neurons on the right here, where you end up with no significant enrichment. So this is a very specific finding in these osteogenic cells. And here's an example of one of the loci, the GDN gene, which is associated with osteoporosis. Obviously, it's all when it has a Mendelian phenotype, it's a skeletal dysplasia, and the variant is right in the middle of this peak, which exists in the 3' UTR of the gene. So this is the model that your osteoporosis risk is in some way influenced by cell fate decisions, and these variants that exist in these loci that are opening up early in osteogenesis are really helping to make that decision for you. So it's a different way of thinking about the pathogenesis of this condition, and it's something that hopefully we'll be able to test experimentally. Okay, now this is the final part of the talk. And this is the part that I think is one of the weirdest and most interesting pieces of preliminary data that we've ever generated. And I don't have answers for you yet, but this is somewhere that I think could be really interesting for a lot of us to pursue. We did this RLEA analysis of de novo variants using 4 cohorts of individuals who are from autism studies, and then the TopMed study, which also identified de novo variants in a bunch of trios. And we asked the question, are these de novo variants enriched for regulatory loci in different cell types? And while there are some differences that you see for the neurotypical MSSNG siblings, it's a very small group, it's only a few hundred individuals, these are much bigger. By and large, what you're seeing here is that de novo variants, and each of us has about 60 to 100 of them, that they're non-randomly targeting loci that are regulatory in derived somatic cells. These are germline events, but somehow they know that they're able to predict where these regulatory loci are going to be in somatic cells, and we target them non-randomly for variation. This is kind of nuts, right? And we now have to try and dig this out. And Cathal Shoiga and Donald O'Shea in Galway, Donald is a graduate student working with Cathal. Cathal is professor of bioinformatics. I think Cathal is the guy who will be able to figure that out with us. So we're going to dump it in his lap to solve, but it's a fascinating observation. So how do we go? Is this something that we should have expected? And I would argue yes, because remember I showed you John Stamatoianopoulos' plot here, where we have the increased nucleotide diversity at transcription factor binding sites. There's probably, these observations are probably consistent with each other. If we've ongoing de novo variants landing in these transcription factor binding sites because of something selective that's happening in our germlines, that could be very much associated with this, obviously setting aside selection effects from evolution. So these loci that we're looking at here, we're used to looking at one cell type, one genotype. What we need to start thinking in terms of, and this is where I always have a great discussion with Chris Glass, is what's the effect of polymorphism in populations? Is it possible that the degree of nucleotide diversity for these transcription factors is so great that we can't have an assumption of a fixed epigenome, that the epigenome is by itself polymorphic, and this could be an insight into disease pathogenesis. So Ryan Hernandez up in UCSF, and this is where I'll leave you, has been looking at lymphoblastoid cell lines and asking the question, what variants are associated with the heritability of gene expression between individuals? So he's basically looking at the variants that are associated with the differences in gene expression between several hundred individuals that he studied. And what you're looking at on the x-axis here is minor allele frequency. So you go 10%, 1%, 1 in 1,000. And you can see that this starts to jump up when you go below 1%, closer to 1 in 1,000. And then you have this inset here where you go to 1 in 10,000 and lower. And basically the variants that are ultra rare, the variants that you don't see in Nomad, because they just, you know, they've never been seen in the 150,000 people or whatever it is that's in Nomad. These are the ones that have the biggest effects. And these are likely to be represented by the sort of de novo variants that we've all accumulated as we were born. So this is our opportunity. If we can understand some of these de novo variants that are ultra rare and how they could be contributing to cellular phenotypes and human disease phenotypes, then we potentially have the ability to understand non-coding variation. So what I would argue is that we can use DNA methylation and some of these other types of assays as a great biomarker. Like I'm really impressed with the work that Aaron Halpern and Noah Zeitlin have been doing at UCLA, where they're showing that methylation risk scores actually outperform polygenic risk scores. So we without knowing the mechanism, we can use this in a very valuable way in terms of population health. But otherwise, I've shown you examples of all of these types of outcomes from these epigenomic assays that will allow us to have better insights into what is actually mediating these pathogenic effects or physiological effects that we're interested in studying, so we can understand the disease. So with that, thanks to my group, my Genome Diver group, which is a medical informatics thing. I've got some great external collaborators, and I'll be happy to answer any questions.
Great. Thanks so much, John. That's incredibly interesting. Yeah, very great. So, so questions from the audience.
Hey, hi, Kyle. Maybe I'll start out.
Sure.
So, John, thank you for a terrific talk. I have to say, I'm not really sure what you mean by derived somatic cell. Could you expand on that a little bit?
Oh, yeah. Okay, that was kind of shorthand, wasn't it? It's the idea that the de novo variant has arisen in spermatogenesis or oogenesis.
Wow.
But we're actually seeing it landing. The de novo variants are present in what will be regulatory elements in derived somatic cells. In other words, once fertilization takes place, you've got the soma. How did it know that this was going to be a regulatory element in the liver or in muscle or in brain? So that's nuts, right? You know, I think there's so many questions to be asked there.
Yeah.
Could
Geographic location? Is it that it's open early and thus available for mutation? Or is there a selection of some sort?
Exactly. But if you think about it, and I do, why would you impose the Kupffer cell attack-seek pattern as well as every other attack-seek open chromatin pattern during spermatogenesis, just so that you'll, you know, it just doesn't make sense.
Yeah, the mutation is happening in a place that's going to be open later.
Exactly,
right.
Exactly.
It's early.
So I would love to have some really good data sets of attack-seek from spermatogenesis, and they exist, but oogenesis is so tough because there's so few cells. So, you know, it may be that the loci that we're seeing overrepresented are quite constitutive. That's one hypothesis. So they are actually open in spermatogenesis and oogenesis, and then they remain open in these other cell types. That's testable if we have the data.
Yeah. Thank you.
Great. Okay, you have a question in chat. If you want to.
Are there any de novo sequences that are relevant arising in transcription factor regulatory regions compared with target sequences? De novo sequences. I'm having difficulty with this.
Do you want to speak your question, AC?
Yeah, that might be a good idea. And if you prefer not to, I can give it a stab, but I'm having difficulty with the target sequences, de novo sequences. I'm not sure what's being conveyed there. I'm sorry. Oh. Well, maybe we can just
try to
skip over that.
Try to rephrase it, and then yeah, maybe come back to that. Other questions? My question, John, on the last part of your talk was, so these de novo variants are enriched in regulatory elements active in a variety of cell types, right? Is that reflect that these mutations are occurring in sites that are active in many different cell types, and that's why you're seeing enrichment across cell types, or is it they tend to hit like distal elements that are active in different cell types, but not necessarily all together? Is that, could that enrichment just be explained by, like, can cell type enrichment be explained by those variants tend to occur in like very shared
constitutive
promoters?
Yeah, that's our guess at the moment, but we're testing it, and if it's constitutive, then it could be that these are also constitutive in gametes, which would allow something to be distinctive about the locus in gametes that would then make it more targetable. There is some literature that I'm beginning to get into, which talks about the increased mutability of certain loci in the genome, some of which is related to transcription factor binding, but there's also this kind of U-shaped relationship to transcription locally. So if you've got a low amount of transcription or a very high amount of transcription, it looks like those loci tend to be more mutagenic, whereas this sort of Goldilocks intermediate level is actually decreased mutagenicity. So I think all of these models are on the table, because obviously if you're a cis-regulatory element, you've got some local transcription enhancer RNAs and so on.
Great. Okay, other questions?
Emma.
Yes, I enjoyed that. Was great, John. Thank you. I thought it was fascinating when you said the methylation risk score outweighs the polygenic risk score, and I was wondering if you could talk a bit more about that, and also how do you go about showing that, validating that the methylation is a better predictor of the risk variance?
Okay, so why it happens may be due to exactly the issues that I was talking about in terms of why DNA methylation observations are kind of confounded. If you're going in there with the idea that it has to be cellular reprogramming, but DNA sequence variation and environmental effects and cell subtype effects can all be influencing your signal. If having cardiovascular disease is associated with certain genotypes, as we know it is, and if it's associated with certain cell subtype changes, and certainly indices of obesity have been causally associated with changes in DNA methylation in peripheral blood, and if what else? Cell subtype effects well, basically all of these things that we're calling confounding effects, if they're actually helping to change the DNA methylation pattern in the peripheral blood to reflect this complex kind of exposure, that may be more information embedded within it than just genotype on its own. That would be my guess. But I think to validate it, we just need more studies, better studies, bigger studies. This is just, old-fashioned statistical genetics.
No, I think it's a great idea, and it makes sense that you're basically putting all of these confounders back, many outputs into a single readout effectively, that they're adding up on the methylation. Yeah, I just interested in how, because we such a focus on polygenic risk scores, right? And how do you integrate mechanistic insight with sort of a more statistical genetics approach to
yeah, absolutely.
Yeah.
Terry, you have your hand up.
Yeah, I want to follow up on Emma's question. I'm thinking about your golden mice and driving them toward agouti. And, when you did that study, which kinds of sequencing, did you use a TAC-seq to look at that? Did you, maybe you could go back and just kind of walk us through the logic of how you figured that out. Yeah,
we didn't do that study. That was work by Emma Whitelaw, I think was the first person who studied this.
Okay.
20-something year old work. They first of all mapped that it was an IAP element. So they showed that there was this local mutation. And then all they did was DNA methylation studies and showed that the DNA methylation was correlated with more brownness of the hair versus yellowness of the hair, and less obesity versus more obesity. So purely DNA methylation, purely at the IAP element.
Okay, good. Really great example of, using your environment, in this case, diet, to alter your epigenome. Yeah, and that's the mothers. That was the thing that was striking there. I wanted to revisit that. Thanks.
But the other thing to remember is that in the mottled animals, it's a mosaicism. So its cells are making choices whether they're going to go fully methylated or fully unmethylated. So it also has this sort of epigenesis Waddingtonian component to it as well, where there's a decision being made whether you're going to go down one versus the other. And then depending on how many you send down one versus another pathway, you'll have more or less yellowness of the animal. So it's fascinating.
Great. Okay. Well, it's 1 o'clock. So unless there are other pressing questions, thank you so much, John. Thanks everyone for the discussion. Yeah, thanks everyone. John, so you have the student session.
Different Zoom link I see, so I'll jump over to
Yeah,
that
I'm gonna
now
post
after
it
this.
here.
She'll be on the schedule as well, I think.
Wonderful. Well, thank you very much for listening and for the questions and feedback.
Yeah,
I'll,
thanks, John. Really enjoyed it. Take care. Thank
take
you.
care.
Bye.
Bye.
Bye.